Welcome, everybody. Thank you all very much for joining us here at ARPA-H for a discussion of the decentralized engineering of cells informed by dynamic evidence, or DECIDE, exploration topic. My name is Darshak Sanghavi. I am a program manager in the Resilient Systems Office here at ARPA-H. And I will be taking the next 30 minutes with both myself and my team to do a brief overview of this exciting new opportunity and exploration topic. And then we'll have some time for questions as well. As a reminder, this session is being recorded, and the recording will be available publicly on the ARPA-H website at the conclusion of this session. Again, thank you all very much for joining. I also did want to point out that I'm joined by a number of our team members here at ARPA-H. We're extremely proud of our technical, regulatory, and business financial support that we have. So I'm joined by a number of our science, engineering, and technical advisors, as well as our business finance manager, and of course, our expert in contracting as well. We'll all be on the call. Well, let me go ahead and get started. So we always at ARPA-H, as we think about the work we're doing, start with our what-if statement. And the what-if statement for the DECIDE exploration topic is, what if more patients with rare diseases could access clinically effective therapies? Now, what does that really mean? And I think as we go into it, I wanted to give a little bit of a narrative about the broad goals of this exploration topic. If we go to the next slide. So we are in an unprecedented era of scientific discovery, as many of you who are watching this session are clearly aware. We've made enormous strides in gene therapy, as well as the understanding of the biological processes that can be corrected to significantly improve and in many cases cure what previously had been life-threatening or disabling conditions in the past. Many of these conditions are actually quite rare. They affect a very small number of individuals. And herein lies the challenge that the DECIDE exploration topic is trying to solve. Let me give one clinical example, but please keep in mind, and if you read our draft solicitation, which is online, it gets into much more detail. But let's talk about one condition, severe combined immunodeficiency, what in decades ago was commonly known as the bubble boy syndrome, which was associated with a significant lack of appropriate function of the immune system. And this was due to a Artemis deficient genetic subtype. Now, because of this condition, these individuals who have this can develop severe opportunistic infections that can be life-threatening, infections that many of us that have normal immune systems never have to worry about. Further, these types of conditions can be much more prevalent in certain marginalized, underserved, or other populations. In this case, this particular variant of SCID impacts about 1 in 65,000 people in the general population, but 1 in 2,000 among individuals of Navajo or Apache descent. The amazing thing about this condition, though, is that researchers, and this is sort of, as I talk about this, think about this as one example of many, but researchers have in fact identified incredible clinical strategies and cell therapies that can actually cure this condition. In other words, that material has been well understood. It's been published in top-tier scientific journals, and one would think that would be enough to actually lead to broad-based treatment and cure of this condition, particularly among these children. This therapy, as we said, it's not only is it curative, but it lasts. It could be a one-time treatment that leads to a lifetime of appropriate immune system functioning. However, here's the problem, is that we broadly have not created the sort of approaches to manufacture and demonstrate cost-effective and safe manufacturing of these small batch therapies in a way that meet the current manufacturing requirements from the Food and Drug Administration. That doesn't mean the FDA is doing anything wrong. They're actually trying to, of course, protect the health of the American people. But in this new era of technology where we can develop treatments that, in fact, affect a very small number of people, we need the science and the technology to appropriately demonstrate that these small batches can be appropriately manufactured in ways that really give reassurance that these treatments will be safe in these sort of smaller manufacturing tranches. And that is at the core of what the DECIDE exploration topic is trying to solve for, which is ultimately, can we develop a new set of approaches and breakthrough technologies that can allow for even small batch production to at least demonstrate safety for, per se, less than 100 doses per year, ultimately to lead to accelerated approval from the FDA, which then ultimately would unlock the ability for reimbursement from Medicaid and other payers. And so we can actually move to a situation where right now these therapies are only offered in clinical settings, philanthropically funded, but then we make it so that it's not only the right thing to do to help these children, but it's also financially the reasonable thing to do because those therapies are paid for. That's simply put the goal of the DECIDE exploration topic. We go to the next slide, please. So this is demonstrating sort of a little bit in a graphic manner the problem that we're trying to solve, that I just tried to give some context through the example of SCID. Academic medical centers cannot economically manufacture these small batch therapies because the significant resources to ensure the quality and consistency comparable with large-scale production. In other words, we currently require manufacturers to use the same processes to demonstrate appropriate consistency and quality, whether you're making 100 doses or 20,000 doses. And so obviously, if you have a large market, you're going to be doing thousands of doses. That's a very different set of economic realities than if you're really looking at small batches. So that's sort of one challenge. As a result, regulators have a very limited number of tools for making those decisions around small batch manufacturing. So as a result, those costs are high, and these therapies, even when they're clinically validated or suspected to be quite impactful, cannot meet that burden from the regulatory standpoint. And finally, then, as a result of lacking the regulatory approval or accelerated approvals, reimbursement cannot really exist, or it's extremely challenging. And so it's simply not feasible. And so, for example, as in the case of those Navajo and Apache children, they may lose access to potentially life-saving therapy because we have not solved this problem. This goes beyond just those individuals affected with SCID. As many of you are aware, thousands of rare pediatric diseases impact tens of thousands of children, and only a minority, estimated less than 5%, actually have FDA-approved treatments. Without sort of a way to solve this, we risk taking an unprecedented era of technological advancement and not ultimately getting those to the people that need those therapies. It is, simply put, an ARPA-H hard problem for us to solve. And so we need to solve for both patients, but obviously for those developers, because without sort of these new processes, we just don't have the right incentives to move forward. How can we solve for this? Next slide, please. Well, the DECIDE exploration topic is focused clearly on this issue, as it says in these slides. And I will also say this slide deck will also be publicly available for download and sharing at the conclusion of this webinar. But the goal, as it says here, is to improve that small batch manufacturing and regulatory decision-making confidence in order to overcome those economic and regulatory barriers to ultimately reach patients. And you can see this sort of projected future state if this exploration topic is successful. The academic medical centers could use variability detection and analytic tools so that we can actually get a sense, even with small batches, that we're safe. And we're going to be starting with small batch autologous cell production with ex vivo genetic transformation. That's sort of the target of the DECIDE exploration topic, to be super clear. We think it's important to focus that effort as we're developing this strategy. Perhaps someday that can be expanded, but that's why this is an exploration topic. We want to show this can be done in one specific case, or at least one specific type of manufacturer. The hope is then regulators will use these kinds of statistical decision-making tools informed by those sort of breakthrough concepts that are being developed around manufacturing to inform a set of guidance that can lead to accelerated approval, and then ultimately working with our payers to ensure we unlock reimbursement. I will say that we are truly benefiting from federal collaboration in a fundamental way. ARPA-H is working hand in glove with our colleagues at CBER, at the FDA, in order to inform this exploration topic. And we have also been working closely with our colleagues at the Center for Medicare and Medicaid Services as well, because I think we can all say we are all aligned with this mission of ensuring that we get safe, effective, and potentially life-changing therapies to children who need them the most. And we've talked about a little bit about the patients who may benefit below, as well as the developers that may benefit from this. So now I want to take a few minutes and walk through just a simple sort of vision again of what we're looking for, and then the technical aims of this project. If we go to the next slide, please. So we pride ourselves in trying to take complicated concepts here at ARPA-H and try to ensure that we break them down in ways that are clear and concise. And this slide, again, just shows what I depicted on the prior two to demonstrate the current state and the challenges where individuals with rare diseases can't access affordable therapy, and then that future state where people with rare diseases can get those. And then here you can get a sense of sort of the approach where the researchers develop a therapy, and then it's pulled all the way through to actually getting to the patients we all want to serve. So we'll go to the next slide, and I'll take a few minutes to review some of our technical aims in this project. So I want to emphasize again, this slide deck is a summary of our draft solicitation, which is currently online and can be accessed via the ARPA-H website about the DECIDE exploration topic. A quick note, we're currently in draft format, meaning we are open to comments and suggestions from the community. So if you are watching this or have thoughts and you'd like to share them with us so that we can improve it, please let us know as soon as possible. That draft solicitation will subsequently be posted with a final solicitation, and then the process to apply or submit those applications to us will begin. In any event, there are two specific objectives that we have laid out for the exploration topic. The first, as you can see here, is understanding the best ways to minimize the variation in batch production, what we're calling process variation modeling. And you can see here, we want to optimize that consistency of decentralized small batch autologous cell production, because we want to understand when there's variability. In other words, when the lots are sort of, there's differences, that sort of indicates challenges around consistency. Where are they coming from? And importantly, what are the markers or the signatures that would suggest that there will be significant variability? In other words, the first objective here is to use innovative or breakthrough technologies that can demonstrate, well, how do we tell, even in small batches, that there's variability? What are those markers? And then are there ways for us to detect those in order to see if there's variation? Ultimately, as you can tell, what this gives us is a set of processes, devices, or an approach that can be scaled to decentralized manufacture from different centers that might be addressing different diseases in this area. So you can see here, the first, identifying those sources of variability is part of the first part here, and then the next part is the unique signature association. There's a lot on this slide, and we've tried to lay out our thinking here. Please keep in mind at ARPA-H, we try to lay out the problem and we try not to say this is exactly how you should solve it. This is why these are considered breakthrough topics. We know what we want in the end. We want the outcome. That's why ARPA-H doesn't give grants. We do contracts. And then we leave it to the creativity of the performer community, all of you listening here, to say, we get it. This is how we're actually going to make that happen. Here is the innovative technology. This is the breakthrough strategy we're going to use. And if you can describe that and execute that, that sort of puts you on the path to a successful application here in this area. Next slide. So we first talked about process variation modeling. Let's please go to the second and final objective of the exploration topic, please. The first part is, well, what are the signals? How are we going to get that data? And then the second part is, okay, let's interpret the data. So the dynamic decision support tools. In other words, we want to inform the minimum number of manufactured lots that are needed to confidently ensure quality and consistency for those low volume therapies. I want to point out, you'll see one of the first bullets, both in Objective 1 and Objective 2, calls out that our colleagues at the FDA will be collaborating with ARPA-H by providing technical guidance to inform the scope, methods, and the desired features and capabilities. This is actually truly helpful because having that guidance from our regulators is critical in actually positioning us for success for the performers once they're accepted into the program. So here's the key. Currently, online, we have all the information about our exploration topic, and we'll send out a link with both the slide deck, this webinar, and our link to Q&A for questions. We are open to feedback on the exploration topic. We then will be posting the full exploration topic final solicitation. Once that is available, our teams will be encouraged to submit a proposal. Finally, we really want folks to be aware of this. We want to cast a wide net to get the best performers for this program. I want to emphasize we have not publicly announced the number of performers at this time yet. All of that information will be in the exploration topic solicitation and draft solicitations that are available. We can go to the final slide, and let's go ahead and open it for questions. And my colleague, Julie Hutchinson, on the line will be assisting with trafficking our questions. I know we may have lost some people because of our technical issues, but Julie, any questions we have that have been submitted, at least in the limited time we have left?
I'm not seeing any that have been typed in, so this would be a great opportunity for anybody that's on the line. You can either type your questions to the right. Oh, I see some coming in now. So one question for you, Darshak, is would you consider funding an allogeneic therapy for Duchenne muscular dystrophy since in this rare disease, autologous therapy is not applicable?
Yep. So this is a great area in which one could submit comments and feedback on our draft solicitation. We wanted to ensure that we focused initially on autologous cell therapy, primarily with the focus on hematopoietic stem cells as an approach that we wanted to sort of work through this new process. If there is a compelling case to be made for why allogeneic therapy for DMD could be something we should consider, again, we would welcome comments on that during this draft solicitation period. Thank you for the question.
The next question we have is, reading the proposal, there seems to be a focus on the gene-modified CD34 cells for optimization around starting material. Is there flexibility in using CD4, CD8 T cells for autologous CAR T?
Thank you. So that's right. So I want to point out again, in our efforts to try and zero in on a specific cell type and process that we could optimize for the decided exploration topic, as I said, we focus on hematopoietic stem cells, in other words, sort of similar to CD34 positive cells as the starting material. The team here is asking, what about using CD4, CD8 for potential CAR T for autologous cell therapy? I think this question the answer here falls into the same category as the prior one, which is that if we can make a compelling case for why this QC could be scalable or sort of more broadly applicable and also include, say, hematopoietic stem cells and ex vivo modification. Remember, we're trying to solve for that small batch therapy issue and do it in a way that is scalable in 2 years. So we want to position ourselves for success. So please submit those comments during this draft solicitation period, and we'd be happy to take a look at them. Thank you for that question as well.
A couple more. Is the expectation of work mainly focused on the QC of the expanded cells for release or optimization, or is optimization of the manufacturing process expected as well for our exploration topic?
Thank you for that question. So here, our goal broadly is to ensure that we focus on the QC, because that is part of the major challenge that we're trying to overcome for regulatory approval. In other words, we know that a therapy is clinically effective. How do we know that it also meets those sort of criteria to ensure that it can be manufactured and administered in a safe manner to patients? So that's sort of the critical part of it. If manufacturing optimization is in the service of improving that sort of lack of, improving process variability and so forth, it can certainly be a contributor to that. Thank you. I think we have time for one more question, Julie.
So solicitation states not submitting drug experiments that will be submitted for IND enabling studies. Does this mean that you're only focused on platform technology rather than specific drug approaches?
So
And just
here,
sorry, one thing we're running out of time, so this is going to be our last question. But for every other question that was submitted, our team will pull it down and respond via FAQ. So just making sure that everyone could hear that as well.
Yep. Actually, for this one, I apologize. I know we're at time right now, and I want to make sure that we take this question also and answer it thoughtfully. So we're going to sort of take this one back as well, and we'll post it in our FAQ as well, if possible. So thank you very much, everybody. Please send your comments to us via our portal, and we really look forward to next steps here. Again, grateful to all of you for your participation. Have a wonderful day.